Okay, now let us talk about the striker control methods. How do we deal with these parasites? For your own information, most of these parasites occur in smallholder farming areas. And I mean, in large scale commercial farms, we may find them, but they may not be causing a lot of damage. So, if you want to successfully deal with the parasitic weed, you need to understand the life cycle so that you can choose the points where you can disrupt the life cycle which we have already described. Now, there are some people who have tried to form herbicides using germination stimulants so that the seed can germinate in the absence of the host, thus exhausting the Striga seed bank. Or you can actually breed crops which do not produce these stimulants. This will cause, I mean, you are not going to allow germination process. Or if there is germination process, you can also breed to reduce attachment or to eliminate it. So you can do breeding, or you can use some form of herbicides to reduce this parasite. The other issue I wanted to explain is the relationship of grain yield, it could be grain yield, versus total Striga dry weight. You can see as you increase the Striga dry weight, you tend to decrease the yield. But there's a special curve. It's known as an exponential decay curve. What this graph is saying is that you need a very small amount of Striga to cause a big effect. So if you have few Striga in your field, don't ignore it. You can get serious damage from seemingly few plants. And when you are going to control the Striga, please note that Striga damage damages the crop before it emerges. And it is quite a challenge because you can't control something which you are not seeing. because when it damages the crop, it will be underground. And also, few plants of Striga can cause serious damage, and you cannot ignore few plants. There's a reference. If you have the time, you can read this Striga conodrum pest conodrum, which is under pest management science. It's the Striga conodrum. It is explaining the difficulties involved in controlling Striga plants. Let us look at the host-parasite interactions. What we call a susceptible genotype is that it is going to support high Striga emergence and give us low yields. The one which expresses tolerance, it can have high Striga emergence but high grain yield. Partial resistance, you have got less Striga emergence and slight grain yield reduction or no grain yield reduction. Resistance, which is equal to immunity, here we've got no Striga emergence, you've got high grain yield. We are yet to reach at resistance, which is equal to immunity. Now, if you wanted to control Striga, probably, let me say this, most farmers really do not bother to control this weed. Why? You'll find that most weeds emerge probably with the crop around November, December. This is when we have our crops emerging in this country, soon after the onset of rains, starting probably mid-November, going towards December, and farmers will start their weeding. But these parasitic weeds won't appear from November, December, January. You won't see these parasitic weeds. You can even go up to February without seeing parasitic weeds in our fields. But towards the end of February, or beginning, towards the end of February, probably that's when you start seeing these weeds. By that time, most farmers will have gone through their weedings. They will have, finished their weeding cycles. That is when you start seeing the Striga, and it will have done its damage. And the farmers, will not have any incentive to do weed control. But if farmers grow a resistant or a tolerant variety, they are assured that they are going to get grain yield. So if they see the parasites towards the end of the weeding periods, they could be encouraged to reduce these parasites. The extension message will make sense because they know they are going to harvest something. But if the crop is already dead, and you are trying to convince these farmers saying that please remove the parasitic weed so that you don't increase the seed in the soil, they won't listen to that path of advice, because they are not going to get anything. And you tell them that after 20 years maybe things will be okay. They are not going to listen to that. So farmers need something which can produce a crop, grain. So the first thing which we should do is to find resistant crop genotypes or tolerant crop genotypes, so that farmers could harvest something. In Zimbabwe, we explored some sorghum cultivars and found that we had SAR29, SAR33, SAR35, SAR37, and SAR16. Around 1996, after evaluations, there was interesting resistance. Then some of these became sources of Striga resistance. And Mtengwa in 2005 crossed SAR29 to SV1 and indicated that the recessive gene controlled the low stimulant production, which he termed the LGS gene. So these SAR sorghums were actually producing less Striga lactose. That was the basis of their resistance. And this could be, passed on in crossbreeding to form some genotypes which could produce less stimulants, and yet they could also produce high yields. There are also some sorghum land races like Ashigisire, Katandanzara, Mutode, Nyamudzi, Terwane, and Igan, which could be tolerant or resistant, as reported by Chanyowedza 1996. Also Mandumbu confirmed some of this information reported by Chanyowedza. Here we've got a paper which was screening some sorghum genotypes for resistance. Maybe you wanted to follow it up. What about maize? Tolerant maize cultivars. Now, after screening quite a number of maize genotypes, it appeared that CML395, which is a CIMIT inbred line, was showing some resistance, or they were actually tolerant to Striga. Then it was interesting to note that R201, which is an old hybrid, that old hybrid was bred specifically for drought, and this one is a drought tolerant genotype, because drought and abscisic acid are associated, and at the same time Striga infection is also associated with abscisic acid. The question was, since R201 is associated with this abscisic acid, and it is resistant to drought, could it not be tolerant or resistant to Striga asiatica? It was one of the interesting questions which we tried to answer. And indeed, we found that R201 was attacked less by Striga asiatica, and it could do well in Striga endemic areas, in places like Chundura communal area, where we find a lot of Striga. We also have IITA maize cultivars, which have been shown to be resistant or tolerant in Tanzania by Mbraga and Masawi. Now we have this question, Striga resistance in maize. For a long time, people, were wondering whether maize could actually be resistant, could resist Striga infection. However, it was noted that Striga resistance actually occurred in wild relatives of maize. For example, in a grass known as Zea diploperennis, it's a wild relative of maize. Then there is also Tripsacum dactyloides. These are grasses with sources of Striga resistance. So what happened at IITA is that they developed a maize line known as ZD05 with the Striga resistance. They took Zea diploperennis and crossed it to tropical germplasm. And then they produced this ZD05 with the result that it was resistant to Striga, and it has now been used to develop genotypes with Striga resistance. This is the Zea diploperennis, a perennial teosinte or wild maize. This is Tripsacum dactyloides, again a source of Striga resistance. So how does ZD05 resist the Striga infection? It could be through avoidance, through less branched root architecture, but this is to a low extent. Then it can resist attachments. If you look at the life cycle of Striga, at the point of attachment, we can interrupt attachment using this ZD05. Again, it doesn't support growth of attached Striga. So we can stop the life cycle, and then probably this variety, this genotype could help us to solve the Striga problem. On these graphs, I just want to show you what we normally do when we want to evaluate Striga tolerance. It's a quick method which can be used. You grow your maize in Striga infected area or pots. The other one should be in an infected one. Then you measure the heights. You then compare the heights. If the heights are separating, it means you have got high susceptibility, like probably here. I don't know whether you are seeing the arrow there. There's a lot of separation here. But if there's little separation, it means there's tolerance. So this is a quick method. And we have used it here in the department to screen quite a number of maize genotypes. Okay, so this is the way you follow the heights. A lot of separation means high susceptibility. You may also have to read some of these papers if you have the time. Reaction to new quality protein maize genotypes to Striga infection. Quite an interesting paper by Nyakurwa, one of our MPhil students. Here I'm showing you the separation of, if you look at these graphs, where there are big gaps between infected and uninfected, it means the genotype is susceptible. Just look for R201. You can't separate the lines, which means R201 is very tolerant to Striga infection. So this is a polythene bag with maize. This is Striga. Again, this is the polythene bag. This is during screening experiments. I think this was an MSc study. Here we are evaluating some maize hybrids for Striga infection in Ngokwe. Again, maize hybrids in Ngokwe. Evaluation of maize hybrids in Ngokwe. Yeah, the person in the background there is a breeder. This is Professor Gasura holding that cob. So here in the department, what happened is that we took some IITA maize hybrids, inbred lines, which were resistant to Striga hemontica, and these were used to develop some hybrids. And one of the MSc students, Wafa, did quite a lot of evaluations of these hybrids. So probably before I proceed, we can say we now have some hybrids in maize with some useful resistance. And these hybrids were produced by our department. We now have a company known as VN Seed Company. Maybe it will produce a number of seeds for market, and some of the seeds, some of the maize seed should be tolerant to Striga asiatica. Tobacco tolerance to Striga gesneroids. It was reported that T66 was the most tolerant to Striga gesneroids, followed by T61 and the KRK26. This was reported by Coca. This is the tobacco affected by Striga gesneroids. You can see it can affect the leaves. But this is a wrong variety in a Striga gesneroids infected area. So Koga was one of our DPhil students who worked on Striga gesneroids and its control strategies in tobacco. So the topic was genetic characterization of Striga gesneroids and its control strategies in tobacco, Nicotiana tobacum. That was his PhD title. Mechanical methods. Yes, we can use mechanical methods, but remember that most of the damage will have been done because we can do them in February. This is the reason why we need to have a crop which resists or which is tolerant, so that we can combine it with the mechanical methods. We can burn the Striga. As far as herbicides is concerned, for a long time on commercial areas, they were relying on 2,4-D and MCPA, which were being applied post-emergence. But we know that during that time, most of the maize will have been damaged, and few farmers may want to use this approach. But yes, it could have worked in commercial areas over a long time. Dicamba can be translocated. If applied to the leaves, it can reach the roots and interfere with the parasite. So dicamba could be an option. This technology, where we have imazapa resistant maize, which was developed by CIMIT, could be used to control Striga. It has two components. You have got herbicide resistant maize seed and imazapa, a systemic imidazolinone herbicide. So you put the herbicide on the seed like a powder. You coat the seed with the required dosage, like for example 30 grams of imazapa per hectare, in order to control the Striga. What I can say is that in Zimbabwe we have not yet developed this technology. But in Tanzania and Kenya, West Africa, they now have this technology. Yeah, I just want to show you, if you don't have Imazipa, you can see these two. There's a big difference in the height of infected and uninfected. If you put Imazapa, you narrow down the growth, the differences. Again, these are the CIMIT line 1 and line 2, which show differences in supporting Striga. We can use crop rotations. We can use trap crops or false horse in the rotations. False horse produce strigalactons, which causes germination. If you look at the life cycle of Striga, where we have germination, we can cause germination, but on the attachment there's going to be failure because some hosts don't support attachment. So we had bambara nuts, BN Dagutangu, Chizanga, Chipata, and so on. And also cowpeas, which were particularly good in reducing Striga seed in the soil by causing a lot of germination. So you can rotate your maize with these with either bambara nuts or cowpeas. Catch cropping, growing of susceptible host, so you grow a susceptible host, then you plow it under. You can reduce the Striga seedbed. You can use Sudan grass, and this can be done in abandoned lands. This can be easily said, but doing it is another issue. Imagine, like now people are now preparing for the cropping season. You come in with an advice saying that some of the fields grow a crop which you are going to destroy. Very few farmers will take up that kind of advice. So yes, catch cropping can reduce the problem, but it's not economically attractive. It cannot pay the farmer during the current season. Intercropping, you can intercrop maize and cowpeas, probably in the same row. It has been found that in the same row, it's very effective in reducing Striga emergence and also increase the grain yields. There's a study which was done by Kasembetar in 1998 which indicated this issue. In 2002, intercropping maize with the Desmodium cynatum greatly reduced the Striga hemontica in Kenya. So intercropping could be useful. in reducing Striga infection. Here I'm just showing you the data from intercropping maize with the various cowpeas. In 1996-97, you can see that wherever there was cowpea, the yields were higher than the sorghum maize. Even in 1997-98, although there were no significant differences, higher maize yields were achieved when there was cowpea. In this case, the cowpea was reducing the impact of the Striga. Nitrogen, most soils which are depleted in nitrogen are the soils where we find the Striga problem. So nitrogen actually reduces the impact of Striga on cereals. The effects of nitrogen depend on crop cultivars. Now, Agboli in 1991 suggested the use of 120 kg per hectare of nitrogen to induce Striga tolerance in maize. This level is just too high for farmers. So we tried to look for a lower rate, but with appropriate genotypes, and found that probably 30 kgs per hectare in combination with the CMR 395 or R201 could solve the problem. In tobacco, 50 kg per hectare nitrogen could reduce the incidence of Striga. So what are the mechanisms? Nitrogen could decrease the number of germinating Striga seeds, or low root exudate production can occur. There can be inhibition of attachment process on the host roots. Number 4, nitrogen can stimulate photosynthetic activity of both parasite and host. Probably this brings about tolerance. Then phosphate can also reduce the incidence of Striga. We have diammonium hydrogen phosphate, which was applied to sorghum. It reduced the production of Striga lactones. For example, sorghumol and 5-deoxystrigol. It also reduced the Striga emontica emergence. So the two elements, nitrogen and phosphorus, can be used to reduce Striga infection in maize. Anyway, these graphs just show that application of phosphate reduced the Striga infection in sorghums. Also here it shows that increasing phosphorus also reduced the Striga incidence in maize. This is the maize with the Striga attachments. The experiment was done also under petri dishes to follow germination of Striga by amending the agar with the different levels of phosphorus. So using 6 maize genotypes, those graphs are actually showing that when phosphorus was increased, either the Striga counts or biomass was reduced. Same information. Well, the other issue is timely planting. Early planted maize had a higher Striga infection, but produced high grain yields. So with early planting, you may not succeed in reducing the population of Striga, but at least the farmer can harvest something. So farmers should be encouraged to plant early in Striga endemic areas, but they should also be encouraged to remove the parasite whenever it emerges. At least they will have something, they will have the yield. Biological control. In West Africa, they've used the Simincronix. It's an insect which can damage Striga montica. We haven't tried it in Zimbabwe. Fusarium isolates were also found to control Striga. Then Abascula mycorrhiza. These ones can actually be useful because they can increase phosphorus production in the root zone of the host, and this phosphorus can actually decrease Striga infection. So a vascular mycorrhiza could be used for the control of Striga in cereals as a biological control measure. Now, what about Electravogeli and Striga gesneroids? We can simply say the life cycle is similar to that of Striga asiatica. And control measures, yes, people can do hand hoeing and pulling, but this may not be effective. But with cowpea, there's a genotype known as B301, which was found to be resistant to Electravogeli and Striga gesneroids. You can have the addition of phosphorus, about 45 kgs per hectare, which can reduce the impact of Electravogeli in cowpeas, as reported by Mugabe in 1983. We think the research focus should now be on screening cowpea and bambara nut for resistance to these parasites. Probably this could become the cost-effective way of dealing with these parasites. You may read this paper. It's about host parasite relations between cowpea and Electravogeli. Also another paper, if you have the time, you may read this. We had one BSc student who looked at the response of cowpea genotypes to witchweed infection. So we were able to see the effects of this weed on cowpea. You can see the leaves are dry, and this was in the pots. Again in the pots. Okay, these are the effects of different, cowpea genotypes which were screened. Now, Cascuta cabestris, this is the spaghetti-like weed. It is a problem in Lucerne, and this is actually a shoot parasite. It also parasitizes tomatoes and carrots, and I think this was observed around the Grasslands Research Station at the Horticultural Research Center. It has little chlorophyll. It is yellow to orange. Here I just want to explain a few things. If you can start with the seed, the germination of the seed requires temperatures which are around 30 to 33 degrees Celsius. This parasite does not require germination stimulants. It just germinates like an ordinary plant. But after germination, it does certain movements at this stage. It can move in a circular anti-clockwise rotation, which is known as circumnutation. It can rotate in search for the host. When it gets to the host, it is going to grow round and round the host, this circumnutation, and forms contact with the host. So it germinates, it forms a long stem which swings around in a circular motion or anti-clockwise motion, which is circumnutation. Then it is going to grow around the stems of its host, forming contacts. At these contact points, it forms hostoria. This is where it can abstract nutrients and water from its host. For its attachment, it uses cutin and some cytokinins so that it is attached to its host. It's now part of the host. Then it forms flowers in the last stages, then produces a seed. For your own information, the seed can stay for up to 50 years, which is quite a long time, and this has implications to control. So this is what I was trying to explain. Of course, it has hard seeds to ensure dormancy, can survive 50 years. Temperature requirements 15 to 39 degrees Celsius. No requirement for stimulants or for germination stimulants. It can emerge and forms a single shoot which can swing in a circular anti-clockwise motion, which is known as circumnutation. Actually, it moves every hour. So it forms coils around the host, forms ostoria, cutiny, forms a bond. Then the ostoria development requires cytokinins. And it is this organ which is used to abstract water and nutrients from the host. Now, what are the parasite effects? It can reduce the growth of the host. It depends on the host for water, mineral salts, and carbon. Photosynthesis in Cascuta is not efficient. Therefore, it needs a lot of carbon from its host. Now, how can you possibly control a shoot parasite, which is part and parcel of the host? You may try hand pulling, probably before attachment, if you can see the parasite, and also pruning of infected plants. Do a little bit of damage to your crop so as to remove the parasite. You can burn the parasite to destroy the seeds. If it is lucerne, mowing can be done. There's also need to identify lucerne which is resistant or tolerant to Cascuta. Probably that could be the solution. Some herbicides like pendimethalin, stomp, and trifluorine can be used. Also gramoxone and glyphosate at low rates, but these may actually kill your host. So it's not easy to control shoot parasites. Now, in conclusion, we may say that parasitic weeds are some of the most important weeds in Zimbabwe, and there is urgent need to look for methods of controlling these parasites. Use of tolerant or resistant genotypes should be the focal point of developing integrated control strategies. We accept that one method is not going to be successful, but you need to integrate methods. But during this integration, the focal point should be use of resistance or tolerance. So no single method is likely to be useful to control parasitic weeds. So here is just a summary. You need to know these parasitic weeds: root parasites, Striga asiatica, Striga forbesii, Striga gesneroides, Electra vogeri, Ramphicappa fistulosa. Shoot parasite, it's one, Cascuta campestris. This is the life cycle. These are the effects. You have to go through them, the effects on the hosts. And this is the summary of control methods: tolerant crops or resistant crops, hand weeding, herbicides, 2,4-DMCPA, dicamba, imazapa plus IR maize, imazapa resistant maize, crop rotation, catch cropping, intercropping, soil fertility management, where you are focusing on nitrogen and phosphorus, biological control, Synchronix, fusarium, and a vascular mycorrhiza. Probably in Zimbabwe, fusarium isolates or a vascular mycorrhiza seem to be within reach. I'm not sure about Synchronix. So this is it on the plant parasites. Let me